Hey everybody, welcome to our next in the lecture series on biomolecules. Today we're going to talk about proteins. Now, I told you that these biomolecules are macromolecules, and they're big, giant molecules, and they are polymers. And so I've been trying to make this connection between, the monomers, the individual subunits that make up polymers, and with carbohydrates, when you think about sugars, you should think about glucose, right? It's a monomer. But I can stick those guys together to make cellulose or glycogen. When you think about lipids, you think about fatty acids, and we kind of combine fatty acids together to make the triacylglyceride or the phosphoglyceride or even cholesterol. That's kind of a stretch with this idea of polymerization, right? So when you think about polymers, I want you to think about a chain of beads. And sometimes those beads can be identical, like in the case of cellulose, right? It's all glucose. Now, today we're going to talk about proteins, and proteins are where this analogy really is profoundly accurate, all right? So today we're going to talk about proteins. And when you think about proteins, I want you to think about a long chain of beads made up of 20 different kinds of beads. So, what are those beads called? Well, proteins are made up of amino acids. Amino acids, all right? Amino acids all have the same basic structure. There's the central carbon or alpha carbon. It's connected to a hydrogen and an R group. You might have seen R already in some of my other diagrams. R is like X in mathematics. R in biochemistry simply means some variable group. All right, and I'll show you some of those variable groups that can be attached to that amino acid. But all amino acids have this group right here, NH2, which sometimes is NH3, or the ammonia group, or amine group. Amine, all right? Hence amino acids, right? So there's the amine group, the NH2. Over here, this is our carboxylic acid. Remember, we saw this attached to fatty acids, right? The carboxyl group, C double bond O, OH, all right? So remember, every amino acid has a central carbon atom attached to a hydrogen, attached to an amine group, and a carboxylic acid. And then remember, I said there's 20 different amino acids, and the difference is that this R group and this R group might be different. Okay. Now here's two amino acids here coming together. Hey, guess how? Well, via dehydration synthesis. So I take an H from here, I take the OH from here. They join together. They involve water, dehydration, or aka condensation reaction, and then I form this very important bond here called the peptide bond between the NH and the C double bond O. All right. Um, Proteins have a variety of functions. The most common type of protein is an enzyme. All right. You can always recognize. Well, this is a funny thing. Whenever I see an IN on the end of some biochemical name, I know that it's going to be a protein, right? Like keratin or insulin or adrenaline or kinesin or hemoglobin. IN goes with protein, protein. Sometimes you might see an EM like collagen, but there's another whole class of proteins called enzymes. In fact, or the large number of genes that you make are enzymes, and they all end in the suffix ASAs. All right, so we have amylases and lipases and proteases and carboxypeptidases. All right, so proteins function as enzymes, and they help catalyze chemical reactions. Proteins make up your body's structure. Some classic uh, proteins that you make up your structure: keratin and collagen. Proteins act as communication molecules, like the hormones insulin or adrenaline or serotonin. Proteins also act as carrier molecules, right? They can be pumps, right? We'll see the NAK ATPase this year. They can carry molecules like the kinesin molecule. They can carry oxygen like hemoglobin does. In fact, here's a representation of hemoglobin here, and it's binding 4 oxygens. All right. Um, over here, I've got the human insulin receptor, and you can have human insulin bind there and send a message to cells to take up protein or take up glucose, or you can have some sort of poison like this cone snail venom. All right, so that's it. So major functions to act as enzymes, as structural proteins, as hormones, or even carrier molecules, and they are polymers of amino acids. Let's look at those amino acids in a little detail here. So remember, the amino acid has a carboxylic acid, an amino group, and then this variable group R here. They come together via the dehydration synthesis to form a peptide bond. Now here's a table of these different R groups, okay? And that R group, this variable group, and I know this is flipped around, but you guys are just going to have to navigate the differences between how they're represented. It all means the same thing. These different R groups make the protein amino acids have different character, all right? So some of these R groups, like the ones that make up the amino acid glycine or alanine or valine, or leucine or methionine, they are nonpolar, all right? So they don't have any charges. They're electronically balanced. Some are polar. They are polar, and they can be negatively charged. Some are polar and positively charged. Some are hydrophobic. Some are hydrophilic. And I wouldn't expect you to know that, but I just want you to know that there's 20 different R groups, which give these 20 different amino acids their different character, their different nature, because that's important in understanding how proteins form their structures, how they fold on themselves. And when we talk about protein structure, we talk about these different levels of structure. The first level of structure is the amino acid sequence, all right? So I'm going to have a chain of these amino acids. Typically, proteins are somewhere between 300 and 3,000 amino acids in length. And the amino acid sequence might be leucine, threonine, isoleucine, proline, methionine, et cetera, down the length of the chain. All right. So that sequence is what we call primary amino acid structure. Now, remember, these different amino acids have different character. Some are polar, some are nonpolar. Remember, oil and water don't mix. So amino acids that have polar side groups don't want to be around amino acids that have nonpolar side groups. Amino acids that are negatively charged might be attracted to amino acids that are positively charged, and positive repels positive, etc. So I can have these little attractions that arise in the primary structure that give rise to something called secondary structure. Now, the classic secondary structures are represented in this globular model as alpha helices. See the helix there, alpha helices, and then these arrow-shaped things are what we call beta-pleated sheets. All right, where there's no arrow or helix, we just have strand. But the strand is still folding in a very specific pattern. In fact, over the 4 billion years of evolution, these proteins have evolved a very, very specific pattern. In fact, if I change one amino acid here, you can throw the whole shape out, right? And we'll talk about why that is as we talk about DNA. So here's a nice conical shape thing. But wait, oh, there's a hole in there. Hey, that's a protein pore. All right. So primary structure is the amino acid sequence. The secondary structure includes these motifs, alpha helices and beta-pleated sheets. The tertiary structure is just the overall three-dimensional structure of the protein. And then last but not least, quaternary structure points to the fact that actually a functional protein will be made up of multiple polypeptide or protein subunits, right? So sometimes proteins can be called polypeptides, right? Because they have many peptide bonds. But this protein here, you'll notice, is made up of like these different colored regions. which are actually different proteins unto themselves. They've come together to form this functional protein. And so that's what we talk about when we talk about quaternary structure. Look, this channel protein is made up of multiple protein subunits. I've got green parts and orange parts. Those are what represents the different parts of the protein, the functional protein itself. Here's another illustration of that, right? This is the protein called kinesin. Kinesin is this kinesin molecule is walking along a protein called tubulin, right? And it's carrying a big secretory vesicle. Every time it takes a step, it burns up some ATP. Here's a representation of kinesin at a more detailed level. Notice that it's made up of these green and blue subunits, and it's got, 12 different pieces of those two proteins, and then it's got this red thing down here that acts as legs, all right? So all those polypeptide chains come together in a quaternary structure to make kinesin. Again, primary structure is amino acid sequence. Secondary structure refers to these motifs, a motif, M-O-T-I-F, the beta sheet, the alpha helix. Tertiary structure refers to the three-dimensional structure of the polypeptide. Quaternary structure refers to the fact that most functional proteins are actually made up of multiple protein subunits. Which brings us finally to our next topic, the central dogma of biology. Where do proteins come from? Well, your parents give you one thing, and that's code for proteins. And the code for proteins is carried on a molecule called DNA. That's right. There's a 1-to-1, well, there's actually a 3-to-1 correlation of DNA to proteins. Your DNA is actually codes for proteins, and that's the topic for our next lecture. See you on the other side.